So hello everyone, I'm Jérôme Burkiewicz. I'm a PhD candidate in the lab of Simon Joly at the University of Montreal, and today I will talk about the first chapter of my dissertation, and this is about disentangling plastic and genetic effect of urbanization on flower morphology in impatiens capensis. So we know that at the macroevolutionary scales, plants face selective pressures on flower traits. And this leads them to evolve difference in morphology, color, scent, and so on. And this is to better fit the pollinator community and also ensure the reproduction. And the outcome of that can be represented on this slide with these two flowers. On the left of the slide, you have impatiens capensis, which evolved toward a pollination generalist. With orange zygomorphic spurred flowers, meaning it can be effectively pollinated by different functional groups, such as bees, bumblebees, hummingbirds, and so on. And on the other hand, we have this lobelia cardinalis to the right, evolving toward more of a specialist. It's mainly pollinated by hummingbirds, and we can see that it evolved very different floral features, such as bright red tubular flowers and a very specific position of the reproductive parts. Now, at the microevolutionary scale this time, the local communities of pollinators can also force plants from a single species to evolve different floral traits. And here is an example of rapid floral trait evolution in Brassica rapa. This is the flower you can see on this slide. And the authors found that it evolved toward more scent compounds when it was exclusively pollinated by bumblebees across many generations. And this is the blue curve you can see here, and to be compared with the end pollination and the surfeit pollination treatment in black and green. We also know that urbanization, the process by which natural areas are converted into cities, disturbs the ecosystems. And this is associated with increase in impervious surfaces, land fragmentation and destruction, and increase in pollution too. And because of that, urbanization impacts organisms too, and particularly the pollinators. So this is a figure from Wenzel and colleagues showing that, generally speaking, the pollinator biodiversity decrease as the level of urbanization increase. And this is represented here by the two red curves. But the exciting thing about urbanization is that we can use it to study evolution in a replicated way. And let me explain this figure. So this is from Santangelo and colleague, and it displays a lot of dots, each of one representing one city for a total of 160 cities around the world. And we can see on this graph that urban areas in orange are very similar to one another in terms of environmental condition compared to rural areas. And because of that, and also the fact that the pollinator communities could be impacted by urbanization, plants could face different selective pressures on their floral traits. And then one question, we can ask one question. Does urbanization impact floral size and floral shape? So to answer this question, I'm studying impatiens capensis, an indigenous species from North America. It's an annual, meaning it can adapt rapidly to changing environments, but also it needs the pollination to persist. Also, impatiens capensis lives in natural areas and remnant natural patches in the city, so it is a great model to study the evolution of flower traits in an urban context. So to study the influence of urbanization on flower shape, we went to the field and we sampled 15 populations in 2021 and 16 in 2022 from the region of Montreal, Quebec, Toronto, and Ottawa. And you can see all the population on this map. So we sampled for each region 2 pairs of urban natural population, and we classify them as such using the normalized difference vegetation index. Basically, it allows us to know if there is a lot of impervious surfaces or a lot of vegetation. And the map represents that with red values associated with the more urban sites and the greener values associated with the sites with more trees. And our urban sites were associated with high proportion of built areas and high proportion of impervious surfaces, and then low value of NDVI. And they were very different from our natural sites, as you can see on this PCA, because our natural sites were more associated with a high proportion of trees, a very low proportion of impervious surfaces, and then high value of NDVI. So in 2021 and 2022, we visited each population of impatiens capensis, and we photographed 50 flowers from different individuals. We also recorded pollinator visits for 4 hours at each site, and we also collected pollinator of impatiens capensis to assess richness, abundance, and size. So for the rest of the talk, I will use the small icon of a forest you can see at the top of the slide to talk about the field experiment. In the field, I also gathered seeds from each population I visited in 2021, and it was to run a common garden experiment. And the idea behind that was to look at the phenotype of the flowers, but without the plastic components of the phenotype. And for the rest of this talk, I will use the small icon of a greenhouse to talk about this experiment. So we then extracted the profile shape of the total data sets using geometric morphometrics. And basically, we did 2 things. First, we placed landmarks on each flower. This is a red dot that you can see, and it allows us to identify homologous points, homologous feature of the flowers for all individuals. And next, we also placed semi-landmarks represented by the blue curves, and it was to capture more complex features of the flowers, like the shape of the posterior sepal and the spur. And we also estimated the size of the flower using the surface area of the posterior sepal. So the first question I wanted to answer was, are impatiens capensis pollinator communities impacted by urbanization? And this is important because different pollinator communities could apply different selective pressures on flower traits. So in this slide, we can see the relative frequency of pollinators visiting impatiens capensis flowers computed from the observation data. And the main result here was that impatiens capensis is mainly pollinated by bumblebees, as they represent 75% of all pollination events. And other pollinators represented a very small proportion of all pollination events. And due to their morphology and also sometimes the behavior they have, we don't think they are very efficient pollinators. So next, using a redundancy analysis on the pollinator community of impatiens capensis, we found that urban areas were more associated with Bombus impatiens, and natural areas were more associated with Bombus vagans. So you can see that on the left side of the slide. So it could potentially lead to different selective pressures on flower traits. But we also know that different species of bumblebees can have the same size and exert the same selective pressures on the flowers. So we decided to compare the size. And urbanization impacted the size of bumblebees pollinating impatiens capensis, but the effect varied depending on the region. And it was due to 2 things. First, to the left of the plot, you can see Montreal and Quebec displaying bigger urban bumblebees compared to natural bumblebees. But this thing was due to 2 different species pollinating in 2 different habitats. For the rightmost part, in Ottawa and Toronto, only 1 species of bumblebee pollinated impatiens capensis. But urbanization didn't have the same effect on their size, with urban bumblebees in Toronto being smaller than the natural ones and no effect in Ottawa. So next, and because of this difference, I wanted to answer if urbanization impacted impatiens capensis floral size. And we found in our common garden experiment that flower size differ between habitats, with urban flowers being bigger than the natural ones. But when we looked at the field data, after accounting for the year of the sampling, urbanization didn't impact flower size at all. And this result showed plasticity of flower size and perhaps counter-gradient variation, meaning that the environmental condition oppose the genetic part of the phenotype, and then we don't see any differences in size at all. And finally, I wanted to know if urbanization impacted impatiens capensis floral shape. So on this slide, you can see the predicted flower shape for natural areas in green and urban areas in gray, and it was computed from the Common Garden experiment. And when accounting for the allometry, urbanization didn't significantly affect floral shape. So we can see some slight differences, but it was not significant. Now, in the wild, after accounting for allometry and the year of sampling, there was a slight effect of urbanization on flower shape, with urban flowers having a shorter spur and a less curved spur. So yes, urbanization did impact flower shape, but this effect was mainly plastic, not genetic, and likely due to abiotic factors. So to summarize, we found that impatiens capensis is mainly pollinated by bumblebees. Next, we found that impatiens capensis pollinators are different in urban and natural habitat, and it was due to two things. Sometimes it was different in the composition, two different species of bumblebees, for example, but also we found some difference in size inside the species of bumblebee. One of our main results was that urbanization did impact flower size, with urban flowers being bigger than the natural one, but we found a lot of plasticity in the field. Flower shape was also affected by urbanization, but this effect was only plastic. And because of all this phenotypic plasticity, the phenotypes coded by the genotype could be masked, and one question stands. Does phenotypic plasticity limit selection and adaptation of floral traits? And I tried to answer this question in my first chapter using some selection gradient. Okay, so this is the end of my presentation. I would like to thank all my collaborators, funding sources, and you for attention, and I will take questions. Thank you. Yeah.
Thank you so much. I think I just missed this because you came in late, but how did you measure the size of the bees?
Oh, so I don't have a slide for this, but I use the intertegular distance. This is the distance between the wings of the insects, and we did that for the 700 specimens collected. Yeah.
Did you by any chance collect pollen?
No, we didn't collect pollen. It's a very interesting thing to do with impatiens capensis, but we need a lot of money to run such an experiment in the field, but definitely collect pollen and look at the efficiency of each pollinators is definitely a way to better understand what is happening here. Yeah.
I know it's a little bit different than the question that you're asking, but do you see differences in reliance on cleistogamous flowers in urban versus natural populations?
So we didn't measure that, but all population we use in this study were population in the sun, or like, if it's not in the sun, it's very bright, you know. And because of that, we limit the number of cleistogamous flowers. But it's a way to control, but no, we didn't. Yeah.
So these were sort of wild populations you came across in the city?
Yeah. Oh, okay, so it's wild population, but impatiens capensis doesn't grow like in the city. It's more in natural patches like forests and parks and so on. So it's urban, but like, you know, quite natural habitat in urban areas.
Did the patch size or population of the flowers differ within or among the?
Yeah, so yeah, there is some differences in patch size, but we try to find like population comparable in terms of size and not small isolated population. So it could happen to have drift, and I will investigate that in my second chapter about the genetics. Yeah.
Did you observe differences in color between cities and?
So that's a great question. I didn't investigate that, but we have data and we can try, I think so.
All right, hi everybody, thank you for coming. My name is Amanda Whitehead. I'm going to be presenting my master's thesis, which is on floral evolution in response to pollinator decline under high levels of nutrients. So pollinator species have been experiencing declines in our global environment, and this is very detrimental to plants that depend upon them. So one impact that this can have on plants is that it reduces pollination services, which thereby results in lower seed production to these plants. So plants will need to adapt in order to persist under these pollinator declines. One way that they could persist under pollinator decline is by extending their floral longevity. So floral longevity is defined as the time in which an individual flower is open and receptive to pollination. But extended floral longevity is when this time period is extended. So this is beneficial for plants because it increases the likelihood of being visited by a pollinator. Although there's a benefit of being visited by a pollinator, there is an associated cost of floral maintenance. So having these flowers open for a longer period of time requires more carbon, specifically in the form of respiration that's needed to maintain the corollas. So if these resources that are required for maintaining flowers are drawn from the same pool as other reproductive functions, such as ovule maturation, this will incur lower seed production. So it's predicted that if these costs of floral maintenance do not outweigh the benefit of an increased likelihood of pollinator visitation, then selection should favor floral longevity under pollinator decline. Although nutrient availability might be able to impact extended floral longevity, so we're seeing higher levels of nutrients in the natural environment, which could actually help this resource limitation by providing plants with more resources. So if a plant has more resources, this could actually reduce the cost of floral maintenance, which may strengthen selection for floral longevity. So this brings me to my research question, which is, does nutrient addition strengthen selection for extended floral longevity in response to pollinator decline? And I chose Lobelia syphilitica as my study species for 3 main reasons. The first being that it's pollinator dependent, it's pollinated by bumblebee species, and bumblebees are declining within Lobelia's range, and that range is eastern North America. And then for my experimental design, I had pollination treatment, which had 2 different levels. So I had a 50% reduction in pollination, and then an ambient pollination treatment level. And then for nutrient treatment, I had an ambient fertilizer treatment, which was essentially just a healthy amount of fertilizers that the plant could use to grow. And then I doubled that treatment to mimic nutrient addition. And then I used a 2x2 factorial design, so I had 4 combos of these treatment levels. So I measured for longevity in seeds per fruit for 5 flowers per plant to estimate selection, and then I collected above ground biomass for a subsample of these plants. And I collected above ground biomass essentially to see if this nutrient treatment worked. So before I get into any of the selection estimates, I need to make sure that my nutrient treatment did work. So in this graph here on the Y, we can see above ground biomass. On the X, I have the 2 pollination treatments, and then high in ambient nutrients. So the nutrient treatment did work. The plants that had higher nutrients had approximately 30% larger above ground biomass. And then I estimated selection, so I have 4 different graphs here for all the treatment combos. And on the Y, I have relative seed production. On the X, I have floral longevity as standardized. So in each of the 4 treatments, I did not see selection at all, as can be seen in the S value, which is the selection differential. So 2 things I want you to notice here. So the first is that there's no selection going on at all. And the second is that there's a pretty strong case for that, as all the S values are pretty similar to one another. And then I went in and looked at my data to see why selection might not be occurring. So we have a similar type of graph here, but seeds per fruit on the Y. So looking at this, I did not see an impact of nutrient addition under pollinator decline, and that did not impact seed production. And then I went in and looked at floral longevity. So there was a significant relationship between pollinators that had reduced pollination, plants that had reduced pollination, and they produced approximately 15% longer floral longevity than plants that did not have the reduced pollination treatment. So that provided some insight into why we did not see evidence of selection. So no, my research question was not supported. So the main takeaway from the study is that even when plants have abundant nutrients, extended floral longevity does not help them maintain seed production, as pollinators decline. So they will need to adapt other traits in order to persist under pollinator decline. So some of these could be taller inflorescence or more vibrant petals, and yeah. And thank you to everybody who made this research possible, and thank you all for listening. In the back there, question? Sorry, that was the first hand I saw.
Are these gynodaceous? Do they have the female flowers or the female plants and then the hermaphrodite?
Yes, they are. So this specific population was mostly females, actually. Yeah. Oh, sorry, hermaphrodite, sorry.
I wonder if you would look at a population that was mostly, or are there populations that have mixed? Well, yeah, I guess we have to have hermaphrodites if you only have females. I wonder if there's any, if you would see that floral longevity, selection for floral longevity only in the female population.
Yeah, perhaps. I mean, my study, of course, didn't look at a mostly female population, but I mean, that would be interesting in a future study to look at that for sure. Yeah, you look into that. Oh, yeah.
Did you the size as well as the weight of the seeds?
Yeah, so, oh, sorry. So that was one of our questions, is maybe does nutrients just make like larger seeds and not like a higher number? So I did calculate that, and the seed weight or like size of it, not the actual number, was the same, so that wasn't a factor. So I did check for that, so yeah, because we were wondering the same thing. Yeah.
I was curious if you saw variation in longevity within the plant.
Like, do you mean like some flowers might have been open for like 10 days and some like 2 days? Yeah, we definitely did. There were some that stayed open for a really long time and some that were done like in 2 days. Essentially, my theory is when you're visited by a pollinator, then you typically, your corollas wilt, and yeah, so that was our main theory, was that those that are visited would wilt and be done, but some that aren't being visited are going to be open for like 14 days, yeah. Oh, is time up? That might be, is that, oh, sorry, I don't know.
Do you have data over time for flower longevity? And do you see, once you start excluding pollinators, that your floral longevity slowly starts to go up? Or is it really an effect of, once you have the exclusion there, they're staying open longer because that was there? I'm curious basically like how plastic the response of being open is not.
Yeah, so I think if I'm getting your question correctly, if the plants that have pollinator exclusion, as soon as you put that bag on, then it's like the longevity extends. Is that essentially the question? Yeah.
What does it look like the ramp up? Is it just like all of a sudden they're open places like they're open for X number of days longer, or is it slowly open more?
Yeah, I mean, I definitely saw with plants that I had the pollinator exclusion on, they definitely had longer floral longevity, and that was pretty evident. And we do know through previous studies that floral longevity is pretty plastic in response to pollinator decline. So yes, I'd say it's a pretty plastic trait. Yeah, in the corner. Sorry. One of my theories that I had is that there might be other factors. So although we reduce the cost of resource limitation, there might be other costs that I can't manipulate. So one of those could be ovule maturation. So when the ovules are older, they just don't produce as much seeds. I also thought that there might not even be a benefit. There's no record in the literature of pollinators benefiting plants, so it's kind of hard to measure the benefit. So I think those two are like my two main theories I have. Yeah. Is that it? Okay. That's it for questions. Thank you very much.